Question 2. DNA holds the genetic information in both prokaryote and eukaryote cells. Describe one organizational difference between prokaryote and eukaryote chromosomal DNA. Okay, so organizational difference. So we're looking for very specific things about how they are organized within the cell. And you know different things about this. But it does say the difference between, so that means you need to give how they are different, okay? So both of them. So what you need to say is that prokaryote is a circular chromosome and eukaryote are linear. Pretty much the only way you can go with this. There isn't anything extra. Okay. Name the substance with which DNA is packaged in eukaryotes. That's histones. Sorry, can I just go back up to this one? One organizational difference between prokaryote and eukaryote chromosomal DNA. You cannot say that one is in a nucleus and the other one is not. Okay, what you're looking for is the DNA itself. So therefore it has to be the arrangement of how that is, not where it is. Okay. Yes, histones for 2. That's easy. Okay, state one location other than the nucleus where DNA is found in eukaryotic cells. Okay, so DNA is found in eukaryotic cells in your specific organelles, which we are pretty sure was taken in ancestral cells a long time ago. So we're looking at mitochondria is my expected answer. Absolutely fine if you went down the road of chloroplast, because definitely in plant cells. And there is one other which is kind of an oddity, but definitely would get the mark. If you went plasmid in yeast, and you'd have to say in yeast, that's totally true as well. Okay, because that's the odd thing about yeast, which lets you use it as a genetic engineering target. Okay, C. During DNA replication, two new daughter strands are synthesized using the original strands as templates. Okay. State why the anti-parallel nature of the DNA molecule results in one of the strands being synthesized in short fragments. Okay, so this is basically the idea that we have, if this is our DNA strand, we've got our 5' and our 3', 3' and our 5'. We open it up from the bottom, peel that apart using helicase to do that. But DNA polymerase can only latch on to this end. So we are only adding to this end. So we're going up the way here, and that's absolutely fine. But this one can only start once you've opened up enough of it to connect on at the top and then read down. Okay, so you're trying to give that information, which is tricky. Okay, so what you want to say is that polymerase can only add to one end, okay, so to the 3' end, and that is running from 5 to 3, okay, and that's your statement. Okay, template DNA, enzymes and ATP are necessary for DNA replication. State one other substance required. Okay, so we've got our template, our enzymes, our ATP. So enzymes covers quite a lot of things because that covers obviously helicase and DNA polymerase and ligase. The other substance we need are DNA nucleotides. And you should just have that as a list that you've worked. Okay. Explain why cells need to carry out DNA replication. This is kind of something that you would have said even in Nat 5. DNA replication means that you have an exact copy of your genetic information to pass on to the next cell. Okay, and that's it. It's what you said in Nat 5. Okay, you need to have exact replication because otherwise your cells won't function correctly. Okay, but you have to give both bits. That you get an exact copy that you can pass on to daughter cells. I've just written something down in my notes that I realised I've not put here. The other substance required for DNA replication, just as a reminder, in here I've said the DNA polymerase latches on, but it can only latch on if you have primers available for it. So you do need primers as well. I expect DNA nucleotides was your answer, but primers perfectly fine. Well, that's question 2.